question for Ladislav that others, maybe Amy might also want to answer, but one of the theories of rescue for GRIN1 dominant negative loss of function is that you could overexpress the protein without risk of causing pathogenic gain of function, because the belief was that it's really the other proteins that are limiting the number of NMDA receptors that are produced, and that there were many more GLUN1 proteins created than are actually needed. But with your research, do you think that raises questions about that theory, where you show that it's the GRIN1, the GLUN1s that are limiting the number that are assembled?
Venholt and Vicini made a similar type of study on granular cell. Is it better? Okay. Made a similar type of study on granular cells showing the opposite. And we don't know whether there are differences in cell types, and that it may happen that in granular cells, which are very special cells compared to cortex, have different type of regulation. We have also additional data from hippocampus. We were trying to continue and did experiments where we tried to overexpress NR1, NR2, and NR2B, NR2A subunits in hippocampal neurons. However, the overexpression did not change the content and currents in use in these cells. I interpret these data at the moment, and very preliminary, that there are steps that regulate the expression of receptors on intracellularly, on the level of endoplasmatic reticulum or cellular, and it is not as simple that it's a random combination of the subunits that are available. Though the data indicate that whether you have more of receptors with 2A or 2B, this is really given by the expression of 2B and 2A subunits in the cell. Is it clear? I tried to do my best.
I have a question for Dr. Yuan, but also GRIN Therapeutics and actually the clinicians and parents, because you showed that 24S hydroxycholesterol has a positive effect on the phenotype of your GRIN2B mouse model. And it is a positive allosteric modulator of the GRIN2B subunit. And I was just looking during your presentation, like, is there like a medicine that, is it a supplement available? And I noticed that the CYP processing of 24S hydroxycholesterol can be inhibited by fluvoxamine, which is actually an SSRI. And I was wondering, is there any interest in fluvoxamine for loss of function GRIN2B patients from GRIN Therapeutics? Is there any clinicians who have experience with this drug? And is there any particular things that stand out in these experiences? Or am I just having a brainwave and it's not?
Yeah. The C550 actually is the two compound for that company. They have another similar compound, I think, I'm not sure in clinical trial or whatever, but unfortunately the company was purchased by the different company. I don't know what is the next step. Maybe, you know, there's a chance to move this forward to the clinical trial on this PAM on the GRIN variant patient. Yeah, that is whatever. So far, I have that information. The one I presented is only 2 compound. It's not moving forward.
No, you need to use the microphone, sorry.
Hi. Can you just say a few words about the role of administration of SAGE 550?
Oh yeah, we did IP injection. We also did chow food, which from our the PK study and also other study, it's doing very well. Once you take, you know, I remember exactly, it's a 3 days treatment can stay over 2 weeks in the brain. So yeah, that pretty good PK study.
Yeah, hi, good morning. Thank you for these beautiful talks. Very interesting. I've got a question for everybody that's working with the GRIN mouse models, because it's striking to see that many of the phenotypes that you see, we also see in GAM kinase mice. And maybe that's not surprising at all. At least I don't find it that surprising, but also the clasping, we can put a coin really on there saying we can put money, this is a mutant or not. So it's there's really a lot of overlap, I think. So I'm wondering, because you, Dr. Yuan, you also showed that you actually looked at the synapse, molecularly, looking at, okay, which proteins are still in the synapse and which are not. Did you by any accident also maybe look at CAM kinase or CAM kinase activity? And how much would it be actually worthwhile, once changes have been seen, to cross the mice, to cross GRIN mutants with CAM kinase mutants and see if we can relieve some of the symptoms? Because I would not be surprised that some of the symptoms that we see actually are through the interaction between those 2 proteins, and not solely caused only by one of the 2. I would like to hear your thoughts on this.
Yeah, I think that's a very good point. The only problem is the funding. This, you know, this animal study is quite expensive. Yeah, I think that is a very, very good point. It's highly likely, yeah. So, do you have any thoughts?
Well, I know that when we did proteomics on like the PSD, of course, the CAM kinase is one of the ones that's also deficient. So when you are lacking the NMDA receptors, you're also lacking the CAM kinase protein. So I mean, I don't know whether you could enhance. It's really a question whether are you thinking that if you have a loss of function that you could offset it with a gain of function, CAM kinase, and then vice versa. Exactly. Could you? Okay, so if there's a loss of function of the variant, I guess the question is whether there's CAM kinase also in the periphery, because I'm wondering in the case of, like, you know, looking at the glucose metabolism, whether all of these things that we know about NMDA receptors in neurons, whether that holds true for other tissues where the NMDA receptors are. Okay. Okay. Okay.
Hi, thanks everybody. I've got a question for Amy Ramsey. You mentioned on your last slide about whether this is experienced with patients. I've got a GRIN1 child who exhibits a lot of what you've discussed with regard to your talk, particularly like oxygen and carbon dioxide. But we've never measured his like glucose or blood sugars or anything like that, and nobody's, one other person has mentioned it to me recently, which made me think, is it something that we should be doing more regularly with GRIN patients, or am I totally oversimplifying it? Like, how do you start to see if this is?
I guess that's my goal. That's my hope, is that if we can establish that it's happening in the mice, it might give clinicians the kind of evidence to go and say, I think this is worth studying in the patients. Because, you know, to just for someone to just say out of the blue, let's just try this, and we're going to ask parents to put glucose monitors on their children and measure their diet and their behavior and all of these things, is kind of out of the blue. And so I think it's just showing it first in the minds might say, you know, maybe we should look at this, and then also what should we be looking at. So I think that it would be worth doing. So it would involve doing a clinical trial, just for the mic is not working. Okay, sorry. Okay. Right, I think I might be rambling, but yes, I do think that it's worth studying in the patients, and of course, I think that this would require a clinician to do. I think actually, probably I could write an IRB and do it, but I would prefer to have a clinician do it. Yeah.
Hi, this is more of a statement rather than a question to you, Dr. Ramsey. I have a GRIN 2B son, and we had like just sort of a series of tests that also measured his blood glucose levels, like sugar in his blood. And he's basically been on the ketogenic diet for the majority of his life. He doesn't get any sugar. He gets a very, very small amount of fruit, but the amount of sugar in his, I can't remember if it was stool or blood, but it was like through the roof.
Okay. Okay.
And it was like so interesting for us, and we're trying to figure out like, where is this coming from? Because he does not get any sugar, but it like couldn't get any higher on the test.
Okay.
Like, for the amount of sugar in his system.
So it does make me wonder if it was urine, because when I saw the very fast clearance of glucose, and also thinking, I don't think it's getting absorbed. It did make me wonder, is it being excreted? So this is the part that I would like to then go back and do the same study, but now measure urine and see whether with, I don't know if it's me. That is fascinating, and actually I would love if people have kind of experiences that they would like to share. I would love to be able to collect this information, and it might help refine what we do and measure. So I feel like probably there's a lot of parents who it's been something that's been measured. They do blood studies, but I think it really needs to be fasting glucose that's measured, because of course when you're eating, it's vary a lot, you know. So, but I really do want to hear these stories. Okay, thanks.
Hi, thank you very much, all the speakers. Wonderful talks. I have a general question, and this is about, in the different models we have seen that beyond the cognitive and motor deficits, they very often exhibit some more vegetative alterations, or circadian rhythms are altered, glucose metabolism. So we are going away from the central nervous system and the synapse in the brain. And my question is about, are you investigating the endocrine system and to see whether these sex differences that you reported, Dr. Bertocchi, or the circadian rhythm that can be modulated also by the neural hypophysis and hypothalamus axis. So it's very open to all of you. If you have some insights, thank you.
Yes, it's really a good question. We didn't monitor yet hormone estrogens, for example, or other kind of hormonal issues by now. But of course, it will be really important to do so. Yeah, also to understand better what are these, what is the cause of this difference, and yeah. We are right now starting to collaborate with some physiologists, cardiophysiologists, because NMDA are expressed also in the heart. also kinase, and we saw some difference also in heart physiology between females and males. And you can check our poster. And yeah, but we are just starting, and the problem is funding. So, but yes, really good question.
Sorry, also in our model, I showed only the results in male animals because I didn't have time to go through the all results, but we have also the female. And the female, it's usually or more variable or the symptoms are really worse, so they perform, I mean, it's more strong, the phenotype in female. And yeah, we are wondering why. We are waiting for the study with the metabolic cages to study the circadian rhythms and link this to the metabolism. And so we are waiting, like everyone, for funding.
Hi. Concerning the circadian rhythm, we saw small changes in circadian rhythm in the point mutation. Concerning other tests, and we were, I don't know, maybe 15 tests which we performed, behavior tests, there were usually differences between females and males. So I will try to yell it. It was lost. Some tests couldn't be performed because of great aggression of males. So in intelligent case, where it was nice to study for females, but it couldn't be performed with males because of their aggression. There were differences, perhaps, and we don't have a clear explanation why this occurs.
Yeah, I'll add a little bit about
yes.
the gender difference.
Sorry, thank you very much all for your talks. It was very interesting. I just wanted to make a little comment because you mentioned the high variability in the females in your tests. Coming from a neuroendocrinologist, I see all of your talks, it's amazing, but for me really females and males can be completely different because of the whole hormonal architecture, starting even from the brain, from the hypothalamus. So in our case, and you will see my talk later, but when we perform, for example, behavioral tests like the one you are doing, we really take into account the estrous states of the female that can really modify the response in any type of behavior test exactly because of the hormones. So maybe if you check this out, it will help you decrease this type of variability that you're having.
Sorry, just briefly, we synchronize the estrus cycle before starting the test, but yeah, this is why I showed only the male, because we are sure about the male tests, but yeah, with female we have to go through it. Thank you for the comment.
Yeah, for the animal model we're working on, gender difference first is the body weight. Body weight reduction mainly in females, not in male. That is one difference. The second difference is PAN scan. PAN scan, some markers has a gender difference. Otherwise, the electrophysiology, immunostaining, major deficit in behavior test, we didn't see any gender difference.
Sorry for making a second question. Is a follow-up. So there are sex differences. We agree with that. And we have this problem also in the lab. We are trying to evaluate some therapeutic strategies. And then we have decided, but I don't know, you can criticize, of course, to take these phenotypes that are conserved between males and females. Does it make sense to you, or you think that those that are specific for females or males will be more interesting, just for debating. Thank you.
I take advantage of the silence and add to that. It would be, I think, very welcome for the scientific community that you guys could decide on what kind of tests and what kind of phenotypes you do capture, and make sure you actually share some of those, especially behavioral tests, etc., so we can compare the studies. Otherwise, I think it's going to be very difficult. But I'll let you answer more on Chavi's question there.
May I? From what I see, from our experiments, the strongest difference was in the learning in intelligent age. This is a nice test. It takes 3 weeks, and I would say that it's difficult to use in pharmacological experiments. We would like to have something which we can see the results immediately, quickly, within a day. And this test is nice, it was reproducible, but it's too slow for any pharmacological studies as it lasts too long.
I think it will be very important that we also hear from the clinicians if they see the same difference, because we might be arguing a nonsense thing for us, the families. So I would like to hear if you see any sex differences in our children, for those of you who are studying them. Sorry.
I just wanted to say that I have only studied the GRIN 1, and there may be differences between GRIN 1, GRIN 2A, 2B, 2D, but I think for the GRIN 1s, I'm not seeing anything that's only evident in one sex and not in the other. In general, things are just more pronounced in one sex, and it's usually for me in the males, but it sounds like there might be in the GRIN 2A, it might be the reverse. So I don't think that there's necessarily a behavior that would only be of interest for one sex. You know, I think that all the studies should be done on both sexes, just powered to be able to detect sex differences. So, yeah.
Then GLUN1 knockdown has really low levels of NMDA receptors. Instead, in our cases, the NMDA receptors are correctly expressed, so they are there. And we saw more hyperactivity, and also the females, they respond with a shorter latency to the stimulus, and they have also more severe seizures, so they also die more for the seizures. But this can also be seen in males, so there is also variability in males and females. Yeah, so it's really difficult to say something else.
Maybe we're going to take your question once we have more clinicians on stage as well. May I ask, before we're going to go and have some sugars, can I ask one question? It seems a lot of sugar metabolism being here going wrong. Would that have a bearing on glycosylation of the NMDA receptors? And I'm asking also the structural people, glycosylation. Because surprisingly, quite a number of the severe mutations involve residues, serine, asparagine, threonine, that are targets of glycosylation. Do we know anything about the glycosylation profiles of these molecules, and could that be another therapeutic approach?
It's such a good idea. There's a researcher in our department who does study this, glycobiology, and he's been helping us with the flow cytometry. So I think it's a very good point. I mean, in the same way that we use glycosylated proteins to look at like type 2 diabetes, why not, right? So I think glycosylated proteins in the blood, but also on NMDA receptors is a great idea. I don't think, I haven't heard anything about it, but certainly they are glycosylated, they're proteins, so it's a really good point.
Okay, before going to bio break, have you ever looked at microbiota in your animals? Related evidently to the glucose
not
household.
yet, but we have collected all the feces, but we should analyze them.
Same.
Okay, then maybe we can talk that, we can facilitate that as well. Yeah. There's, okay, one sec.
As the clinician, I'm like in the Netherlands, and I think maybe also in other parts of Europe, like all the children with neurodevelopmental delays will have a metabolic basic screening, which includes screening for CGA abnormalities. And I can say for our cohort of 90, we haven't heard back of any abnormal metabolic screens. Of course, this doesn't include all these, you know, you have CDGs that doesn't affect these assays, so these are the basic screens, but yeah. And just to say on the female-male differences, again, we need much bigger cohorts to evaluate that. I can say for some of the phenotypes we're looking at, we don't see female-male differences, but of course it is over 3 different isoforms. So, I mean, we need to collaborate and make the N bigger.
Wonderful, thank you very much. And there was one question online, but that's going to be answered very quickly about whether the presentations will be accessible afterwards. You may have a better update on that, but yes, in theory, all presentations will be available online after the meeting. So. Exactly, and we hope, and you can always, of course, eliminate sensitive slides from that, so that you have that opportunity, but we'd rather get the full set, evidently. Okay, thank you very much.